All right, in this video, we are going to continue our lecture from Friday, where we are talking about DNA. So let's talk about some mutations. And the first vocab that we want to talk about is intercalating here. All right, so if you have intercalation in your DNA, this could cause a mutation. And intercalation is being shown right here. So that means you have a molecule in between bases. Now, the type of molecule that's going to do this are usually carbon rings, right? So let's just draw our ring-like structure. Not the greatest, but the reason is base stacking. Remember, base stacking is between the bases, and it's a strongish force. And if we have a molecule that can base stack with our DNA, it will. You will increase the distance between your base pairs, and this DNA becomes distorted. And when you try to replicate distorted DNA, you might have an insertion or deletion of a nucleotide. Now, this doesn't mean it will cause a mutation. It might, because mutations are more or less random. And a mutation might not have any consequences. So not all mutations have phenotypical consequences. Remember, a phenotype is more or less something you see, right? Me having blue-green eyes would be my phenotype for eye color. And the reason why a mutation doesn't have to lead to any phenotype consequences, one, not all DNA codes for stuff. So if you hit a mutation in a non-coding DNA, that's usually fine. Two, the codons have redundancy, right? I think there are like 6 or 7 codons that code for leucine. So if you mutate to another codon, so you can mutate to same codon, then the protein isn't really changed. Well, the protein isn't changed, so there's no phenotypical consequence and may not affect protein, right? You could have the same structure. If you remember when we talked about the proteins, we were looking at how different proteins have changed through different organisms. And we're looking at what residues are conserved, which ones are not conserved. If you mutate to a non-conserved residue, that might not matter. You could just still have a functional protein. However, cancers are caused by carcinogens. And a carcinogen is basically something that causes a mutation. So let's look at these carcinogens here. We have a lot of different chemicals, all right? 80,000. This slide was made a couple years ago, so that number's probably increased. And we make about 1,000 new chemicals every year for different purposes like industry. And we want to know what are the dangers of these chemicals. So usually you get a rat and a mouse and you want to check it for cancer. So you feed a rat and a mouse, rat or a mouse, this chemical, and you just check what happens. However, time consuming, very expensive. So not many things do this, not many chemicals, because you got 1,000 of them every year. People aren't going to spend the time and money to do that, unfortunately, since we are checking to see if these will cause a human cancer. Instead, we use what's called the Ames test. So we use bacteria. So what's happening is that we have salmonella and they are His minus. So this means they can't produce histidine. And then we put them on a plate. That's what's going on right here. And we just ask, do you mutate to regain your histidine function? So this plate is also His minus. And 2 days, 37. You also have rat liver proteins here. That's to kind of mimic our liver because our liver gets rid of toxins. So we're gonna say, you have the proteins of a liver in there. And basically, the more colonies you get, the greater mutation. So the way this works is that you have low concentration of your carcinogen on the outsides, very high here, and then it's a gradient from low to high. And what we see when it's low, we in this specific image, we have very little colonies, very few colonies, a lot of colonies here, which means at this concentration, there's a lot of mutation, right? A lot of colonies have gotten that his back. Then at very high, this is too high of a concentration, probably way too many mutations, which can lead to death. Way too many mutations. Let's just do it like that. Um, so that's the quick and dirty way to do this. So we can say, yeah, this concentration, maybe not as much of a risk. At this concentration in the middle, yeah, you don't want to be exposed to that. There is correlation, right? 80% correlation with animal tests. Um, and this is one of 8 tests under the Pesticide Act, one of 6 under the Toxic Substance Control Act. And if this was class, I would ask about the limitations, right? So limitations is obviously bacteria, right? This is not eukaryota, right? This is prokaryota, slightly different systems. Um, Another mutation, you're using liver proteins, not the actual liver, not an actual system itself. 2 days, that can be sort of a limitation as well. Maybe this take a little bit longer to have cancer show up. But yeah, quick and dirty. So we're going to talk a little bit more about DNA here. So what happens when you are exposed to UV light? Let me just organize my notes here. I'm looking for page 4. Where are you, page 4? There you are, page 4. All right. So UV light causes cyclobutane pyrimidine dimers. Glad I got my notes out because I would not have said cyclobutane. Cyclobutane pyrimidine dimers. Basically, these are TT dimers. What happens if you have two thymines that are base stacking with each other? If you hit it with UV light, these will cyclize. And here's a cycle, and you also have a 6,4 here. When that happens, you have a kink in your DNA. Now, we can possibly repair this kink, but sometimes your machinery gets stuck there, right? And then we more or less, once it gets stuck, we have to guess what nucleotides were there. And you can have a mutation when that happens. And if your mutation is cancerous, you can get skin cancer. So that's why it's important to wear your sunblock because this will happen, especially the lighter skin you are, the more this happens. I know that very well as I'm covered in freckles, which more or less arise from that. Now, you can also get hit with X-rays or gamma rays, ionizing radiation. What happens there? Well, one, it can cause your sugar ring to open, which is no good. Then cause your base to fragment. That is, your base just gets blown away. Again, not good because we need to read those. You can also break covalent bonds. Not good. X-rays and gamma rays are very high energy. right? High energy enough, like I said, to break covalent bonds. So, one example of the consequences of this, end of World War II, dropping the atomic bomb on Hiroshima and Nagasaki. Uh, the thing that killed the most people was not the initial explosion. It was all the radiation. And this showed up decades later in the population, um, where their DNA got cut open. You didn't really feel anything, but you just had a much higher rate of cancers, and they also had much higher rate of birth defects afterwards. Right. So that's why it's very important when you go to the dentist or whatever, make sure you're wearing that lead vest. X-rays, especially gamma rays, can do a lot of damage to your DNA. How about oxidative damage? What's happening there? So ROSs, so reactive oxygen species, ROS, H2O2, hydrogen peroxide can do this. Hydroxyl radicals, so that's OH with a free electron. So let's just describe that. Anytime you hear the word radical, and when it comes to biochemistry, that just means extra unpaired electron. Generally, you don't want molecules with an unpaired electron because that is high energy, unstable. And so what will happen with these free radicals, they will give the electron to DNA, or can happen to proteins too, but give that electron away. And this can break covalent bonds because that electron's very high energy. And as we just saw in the previous slide, you break those covalent bonds, that could lead to a mutation because when you are reading your DNA, your machinery will get stuck. And there's ways to get around being stuck. But at the end of the day, if you are stuck, your last effort is just to guess what the base was. So you got a 25% chance of getting it right. And if you don't, you have a mutation. Now, since these radicals are so high energy and sorry, 1 second. Sorry, I had to sneeze there. Since these are so high energy, your system, your cells have a lot of defenses against it. Um, and this is also the reason why, or at least this is a theory why as to we age, right? So your mitochondria, that's where you're doing oxidative phosphorylation. That's where you're making your ATP. That's where you also create a lot of ROSs. And so your mitochondria will break down over time because your cells aren't perfect at catching the ROSs. And this is one of the reasons why we think we age, which is also being shown here and then the middle here. And so one theory is that if you eat less, and you have to eat a lot less, like 1,200 calories a day. If you eat less, you produce lower ROSs and you live longer. And this has been shown in mice, right? You can make a mouse live super long if you give it roughly 2/3 of the amount of food it's supposed to have. Now, again, not tested in humans. 1,200 calories is not a lot, so I would not suggest you try this because that does not seem like a fun way to live. But let's zoom in here. Why are we so zoomed out? But that is the current theory here. All right, so we talked about mutations in DNA. Let's talk about some modifications. One is methylation. Remember, a methyl group is just a CH3. So groups that are going to be methylated are A and C more than G and T. And the type of enzyme, excuse me, sorry, my voice is going a little weird there. The type of enzyme that adds methyls are called methylases. And the donor is this S-adenosylmethionine. I think that's often called SAM. So here is how is the molecule. Look, there's a CH3 right there that we use to donate to add methyl groups to our DNA. Now, we have quite a lot of methylated DNA. 5% of cysteine residues. There's actually CPG islands. You might have heard that about that in biology. So this is just an area of your genome that goes CG, 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 CG over and over and over again. You have a lot of methylation that's happening here, and that's for control of which sequences will be transcribed. All right, so it affects DNA metabolism and gene expression. All right, but let's talk about one of the biggest discoveries about DNA in like the last 40 years, and that's the polymerase chain reaction. So PCR is a method of amplifying DNA sequences of interest. And it relies on DNA polymerase. So anytime we see the word like ASE, that means enzyme basically. So polymerase will synthesize DNA using our DNTPs. So, oh, sorry, one second. So DNTPs, the D is for deoxyribonucleotide. So the N is nucleotides, triphosphates. So deoxynucleotide triphosphate. So this would be like DATP, CTP, PTP, GTP. Basically our DNA bases with three phosphates on them. And you're going to always add to the 3' end. And the things you add on are called primers. And we're going to talk about how PCR works in a second. I always like to bring up Terry Mullis because he's the one who came up with PCR. He said he did it while on LSD driving in California, which is a wild statement to make in your autobiography, but sure. Um, and he won the Nobel Prize for it. But just because you won the Nobel Prize doesn't mean that you know everything. So he was not a supporter of climate change. He didn't think that the ozone was going away for some reason. Big fan of astrology, which is basically I look at the stars and I can tell the future. Um, he thought AIDS is not real and just a theory to get money, right? Um, so great that he came up with this, but a very complicated figure. However, let's look at how PCR works. So those of you who took lab with me this semester, we did this with our corn lab. So let's say I want to amplify a specific region. So I'll take my DNA and I'll heat it up. Heat will separate or remember denature. And to the sample, I have synthetic DNA oligonucleotide primers. So my primers, I have a forward primer and a reverse primer. What these do, what your primers do, they more or less mark the region to be amplified. Right? So let's look at where our primers are. We have one primer that starts right here, one primer that starts right there, right? Then we have thermal stable Taq DNA polymerase. So this is just an Basically, it's a polymerase found from an organism that lives in a hot spring. So this polymerase does not denature under these high temperatures. And so the polymerase will go to the primers and replicate the DNA. But we just want to replicate the dark blue region. So after one step, we haven't done that yet. But if you do step two, we have, hey, we made two copies. Yay. Then you do it again, and we have 1, 2, 3, 4, 5, 6, 7, 8 copies of the DNA that we want. After 20 times, you get up to like 10 million. So from one DNA, in essence, you can go to 10 million DNA. Sorry, keep on coughing there. And this table kind of walks you through what's going on here. So we have number of cycles. So go through 1 through 10. How many DNAs you're making, how many DNAs are not what you want, and how many DNAs you want. So this is your target. So this is good DNA. This is kind of like junk DNA. So after one cycle, you doubled your DNA, but none of it's your target. Go all the way up to 10 cycles, you made 1,024 DNA molecules. 20 of these are not your target, but 1,004 are. So this is the power of PCR. If I just have a little amount of DNA, I can easily replicate it by doing a cycle, and a machine will do this for you, and you get millions and millions of copies of your DNA. So you are amplifying it. So if I have a sample of DNA, I can amplify just one molecule out of that sample. However, that's not the only use of PCR has a lot of uses as shown here. So if you find some DNA from a long time ago, you can easily amplify it and see what it says. You can look at evolution doing something like this. Forensics, if I find some DNA at a crime scene, I can amplify it and I can read it. Viral infections and cancers, right? So if I have some viral DNA in my blood, I can take a look at that and amplify it and see, oh, I'm going to be sick soon. Or the same thing, if I have some cancerous, if I have one of my cells that have mutated and I have some of that DNA flying around in my bloodstream, just take a draw of my blood, amplify for PCR and see, oh, you have some DNA that have some mutations here. Also, we can look at genetic diseases prenatally. Again, just take some DNA of fetus, amplify it, and then do a genetic screen. Um, because once you have a small amount, you can amplify it as many times as you want. You'll just have a ton, right? So here's an example of a Nature paper that was published 2 years ago. In Greenland, they found 2 million-year-old DNA. So they've amplified it using PCR so they can study it. And this part down here has to do with the O.J. Simpson trial. Basically, they found 108 pieces of DNA of OJ's DNA at the crime scene, 61 drops of his blood at the crime scene. Using PCR, they said it was OJ, but this was like 1991, so no one knew what PCR was or really DNA. And so the jury didn't buy it, that was a thing. Anyways, that's just some history of PCR though. So once I have DNA, how do I sequence it, right? We just said I can use it for evolution. We just said that I can diagnose it for any diseases, but how do we actually do that? So let's look at how I can automate DNA sequencing. So the way this technique works is that if I have DNA, I'll have a primer. And then, so I have my DNA that I want to sequence. I also need a primer to start it off. Usually what happens is that you will have a plate, and on this plate you'll have like DNA that you know the sequence of, and then you will attach your unknown sequence. And then you will have a primer. This primer will be to the sequence you already know that's attached to your plate. And then to this, excuse me, to this apparatus, you're going to be adding a couple of things. So your normal DNTP, so remember, the N stands for like all the nucleotides, so this would be A, T, C, or G. And then you would have what are called DDNTPs. So this is like double deoxy. So DD, so in DNTPs, let's just go there first. Remember, the 2' has been replaced with a hydrogen instead of a hydroxyl. To DD, the 2' has been replaced, but also the 3' has been replaced. So when you have DDNTPs, since the 3' is replaced, you cannot add more DNA or more nucleotides to this, right? It's like a stop sign. Now, on these DDNTPs, we also have fluorescence. We have a fluorescence marker. And so each nucleotide has a different fluorescence. So like, I guess if I'm to follow the picture, T is red, A is green, C is blue, yellow is G. So the idea here, if I wanted to sequence DNA, and you would have like 1,000 of these, like 1,000 of these known DNAs on here. So you would do a lot of reads of this same sequence. I would add all my DNAs, both the DDNTPs and the DDNTPs, to this plate. And then I would sort them by size. Once I allowed this reaction to go, I would sort them by size. And then I would just run these through a laser, right? So, for example, this would be low number of residues. This would be high number of residues. So let's say the first read I got, I don't know, let's make it simple. I got 2 nucleotides in before I hit a DDNTP. All right. So 2 nucleotides in and I see C. Okay. So if I see a C, that means, oh, my target that I'm trying to read. Let me get my different pen. So this is, let's do target. If I got a C, my target must have been a G. All right. So in the next read, so let's say I got 3 nucleotides in till I hit a DDNTP, and I'm seeing C again. So that must mean, okay, my DNA is GG. My next nucleotide that I read, that is 4 residue long. Well, that's a T. So that means my target's GGAA. Right. So by having this automated process and this idea of DDNTPs, I can sequence this DNA and then just read the fluorescence I have based on the length of that DNA and the fluorescence I see. I can label my DNA, or I know what my DNA sequence is. And you get a reading out like this with the lines tell you the confidence. So the higher the peak, the more confident you are that you saw that signal. And so if we were in class, I would be asking you these questions, just check your understanding. So why don't you take a second and do that? Pause the video and check your understanding. Um, but I'm gonna go on because I feel my voice going here. So first off, what does the D in DDNTB stand for? Um, More or less double deoxy. So what makes it suitable to be a terminator? No 3' OH. You cannot add on to it. Why do you think this sequence only works for a few hundred nucleotides? That is, why can't we do larger? So yeah, let me mention that. Doing this sequence, you might get like 300 to 400 nucleotides long, but usually not more than that. And the reason is chance, right? This stops when you add a DDNTP. That particular sequencing event stops. And adding a DDNTP is random. So it is very unlikely to sequence a large number of nucleotides without adding a DDNTP. And so by the time you hit like 400, you might be only getting a couple DNAs that went that far. Like you can't do 1,000. You would add a DDNTP way before 1,000. Right? You might add a DDNTP at residue 350, and that runs over. And so if I do 10,000 runs, there's no way I'm going to hit 1,000 without adding a DDNTP before that. So that's why I can only sequence short sequences using this method. But we do have other ways to sequence DNA. So we're just going to talk about one of them. And this is by Illumina. And so with Illumina sequencing, what you're doing is that you're adding 4 nucleotides at the same time, and they are all have different fluorescent colors. So red is C, A is blue. Yeah, C is red, A is blue, T is yellow, G is green. And so what you do, if you, let's say I'm trying to sequence my genome, I'm going to chop that into small bits, into small parts. And then I'm going to add those onto a plate, right? So I'm going to have little wells, each one having a little piece of my DNA, so on and so forth. I then am going to add my 4 nucleotides. Remember, each of these have fluorescence. And the trick here is that only 1 nucleotide adds at a time. They have it set up so their method cannot add more than 1 nucleotide. And then I will measure fluorescence and see where on this, for each mark, what fluorescence do I see? And that's what you're seeing here. That's an actual reading. That's a plate. And I'll record that in a computer. And then I'm going to repeat until my whole DNA is sequenced in each well, right? So I'll just get a recording of like what the DNA was based on the fluorescence. I can then use a computer to add the sequences back together. And I have a complete reading of my genome, right? So we can now read genomes in like a day using this method, where before it took years for the first human genome to be sequenced, and it took millions of dollars. So this is like $1,000. So very simple to do. But I think we will call it here because my voice is going. This will be the last slide for our exam. We will have a new PowerPoint for next week, Monday, covering lipids very quickly. That will be on your final exam. But if you have any questions, let me know. Otherwise, have a good break and see you next week.